So, good afternoon, guys. So, for today's experiment in this lab, the aim of this experiment is to measure and analyze the neural responses in the visual pathway using visual evoke potential. Okay. So, here, first thing in the introduction, we need to know is what is visual evoke potential. As from the name we can analyze that visual means something which we can visualize, evoke means generated from the visual image, and potential, obviously potential is the generation, means anything in the human body is electrically related with biochemical signals. So when biochemically some reaction happen, it will generate a potential in the body. So by visualizing a biochemical reaction occur, and from that reaction a potential develop and for today's experiment we are going to measure use this potential for our test. So firstly, it is a non-invasive test, and it measures the electrical activity in the brain in response to the visual stimuli. Visual stimuli means any picture or any patterns that we can see. And here is the image of the VEP test device. As you can see, there are some monitors, some CPU, some system is there. Second, the purpose of this test is to access the function and integrity of the visual pathways. As we all know, there is a visual pathways which connects our eyes to the brain. So we from this test can help and access the function and integrity of this visual pathways. Visual pathways, it starts from the retina of our eyes and ends at the visual cortex, which lies right behind above our hindbrain. Okay, so there is some electrode placement for this experiment. Here in the diagram, as we can see, I marked three position. First is CZ, OZ, and third is FZ. Okay, this why how could I place my electrode in such a position? All of you know about the international 1020 lead electrode system of EEG? Everybody know? Okay, so on the basis of that 1020 electrode system of EEG placement, I we use the placement parameters like CZ. For the cerebral part we using C, and Z is defining axis here. Similarly, F is for the frontal lobe, and OZ is occipital. Okay, so here we uses three electrodes. So the in this the electrode captures the brain signal and transfer them to the system for further analysis. There are three electrodes. For the placement of electrodes, we need to place a ground electrode. So the ground electrode is placed on the CZ. CZ is right here in the above point. And second, reference electrode at the FZ, at the frontal lobe. And third, the active electrode, which actually records our signals, is placed at the OZ, occipital position. So next is how it actually works. Here I can try to visualize and try to explain you with this simple diagram that there is a checkboard pattern. This checkboard pattern, our both eyes can visualize it. But as you can know, our left eye is coordinated by right brain and right eye is coordinated by left brain. So by this by understanding this thing, we place our electrode right in between so that we got both signals. As we can from this in this experiment, we try both left for left eye and alone single, and for right eye also. So for that we have to place in between to get both signal at the same time. And here is the circuital diagram that OZ and CZ. CZ is positively placed and OZ is negatively placed. Because of that reason, we get a reversal pattern. The peak there is a depth, and then there is a inclination on the peak. The amplitude of the inclination is roughly around 5 to 10 microvolt, and the peak we got is at n75 and n. Okay, next slide I can show you. And there is a latency delay which is around 100 to 110 milliseconds. Here what there are types of VP tests that are performed in a which are pattern reversal VP, which includes a checkboard pattern. The pattern will switch. Sometimes black goes white, white goes black. This type of pattern reversal. Why the checkboard pattern is being used? Because our cortex is sensitive to edge and contrast, and we have a black and white contrast right here. Second is a flash VP. Flash VP means a bright light and suddenly a flash of light and suddenly darkness. So in that case, why we use? Because our occipital cortex is relatively insensitive to flash. So by using this, there is a third one, which is steady state VP, means in which we are producing some continuous stimuli to our brain, and on the basis of stimuli we can measure how our brain reacts. The equipments that we are need to require is the visual stimulus producing device. Second is the scalp electrodes. Third is amplifier, and fourth is a computer read in and read out system. There are some prerequisites that we need to follow. There is a no distraction, sound and light that need to be there. Second, the scalp should be cleaned for proper taken of the signal from brains to the electrode. And the third is the electrode should be firmly placed on the scalp so that no noise signal may be generated by displacement of the electrodes. Here is a waveform, VEP waveform, that be generated by this experiment. Here are some peaks that you can see is N95, P100 and N145. These peaks are actually being used to measure the delay and amplitude. On the basis of these peaks and crust, we can measure. So N90, N75 for 75 millisecond, P100 for 100 millisecond, and N145 for 145 millisecond. Here are what are the factors that affect a VEP test? First, the size of the stimulus. As the size of the stimulus decreases, the increase in the amplitude will occur. Second is the position of the electrode being placed on the scalp. As I already told, if we don't place in the middle line, then it will be taking more left side. If it is being left tilted, then it will take a more left signal as compared to right. Similarly, if it is more right centered, then it will take a more right signal than left. Third is age. Obviously, with the age, the amplitude of the visual stimulus may affect, and it will be decreased. And the fourth is the attention of the patient. Patient need to be very attentive in this test. If he or she will change his focus, then we can't get the proper waveform just like this. It may be distorted, and distortion will lead to wrong analysis. Here are the application why we can use it for diagnosing multiple sclerosis, detecting optical nerve issues, accessing visual pathways, and monitoring neurodegenerative conditions. Okay, so these are my references, and thank you guys.